Hi everyone, I'm Duleepa Pathiraja from the Computational and Synthetic Biology Laboratory, Korea University. Today I'm going to present a recently published article for our Journal Club meeting. The title of this article is Quantitative Analysis of tRNA Abundance and Modifications by Nanopore RNA Sequencing. This article was published in Nature Biotechnology in April 2023. The research group is from the Center for Genomic Regulation, Barcelona Institute of Science and Technology, Barcelona, Spain. To start my presentation, let me talk some background information. As you all know, tRNAs are small non-coding RNA which play a central role in the protein translation, which provides the link between mRNA and protein. And tRNA is usually 76 to 90 base pair long, and this tRNA has self-complementary regions which allows the formation of characteristic structure which is known as cloverleaf structure. Based on their amino acylation identity, there are 20 different types of groups which are known as allo acceptors. For each allo acceptor group, there are several tRNA that translate synonymous codon with the same amino acid, which are known as isoacceptors. tRNA structure is modified by tRNA modifying enzymes, and these enzymes are present in all domains of life. There are diverse types of chemical modifications including methylations, acylations, isomerizations, deaminations, and conjugation to amino acids. And certain tRNA modifications are found only in single tRNA species. For example, 2-lycetine tRNA modifications are found only at 34th position of isoleucine tRNA with AUA anticodon. On the other hand, some modifications are common. For example, pseudoiridine can be placed at diverse positions in multiple isoacceptors. The figure in this slide summarizes commonly found tRNA modifications in tRNA molecules. tRNA modifications can sometimes function as identity elements recognized by amino acyl tRNA synthetases. These modifications affect the decoding preference of tRNA molecules and thereby restricting or increasing the wobbling capacity, which means prefers one type of isoacceptor during the translation process. As shown in the figure, tRNA modifications are dynamic depending on the environmental signals and allows the use of preferred codons under certain conditions. So any deregulation in tRNA modifications can cause some diseases or disorders as well. If I further summarize the content, tRNA modifications are regulated upon environmental exposures, and tRNA abundances are subjected to fluctuations upon environmental exposures. Now, therefore, modulation of tRNA abundances and tRNA modifications are molecular strategies to adapt to certain physiological conditions. So the next question is how to quantify these tRNA modifications and tRNA relative abundances. There are several analytical techniques to analyze tRNA abundances and modifications. LCMS or liquid chromatography mass spectrometry is one of the most sensitive methods to quantify tRNA, even as low as femtomolar ranges. But this method cannot identify the tRNA isoacceptor that contain the detected mutation, as tRNAs are fragmented before the analysis during this process, and it is impossible to track the origin of the analyzed fragments. The other method is NGS based methods, and these NGS based methods are also employed to quantify tRNA abundance and detect tRNA modifications. Even though there is a substantial improvement in NGS based tRNA sequencing methods, these methods still have some drawbacks. For example, tRNA should be converted to cDNA before sequencing, so this leads to erasing tRNA modifications during the reverse transcription. And also, NGS based methods introduce significant bias during the library preparation and incomplete reverse transcriptions due to the secondary structure of the tRNA molecules, and incomplete demethylations and incomplete PCR amplifications. More recently, nanopore sequencing was used to directly sequence native tRNA, and this method was initially developed for sequencing native mRNA. In this method, RNA molecule is directly sequenced, so any nucleotide modifications can be captured, unlike the previous methods. So first mRNA are captured and reverse transcription adapter is ligated. Then there is an optional reverse transcription step to linearize native RNA, and then sequencing adapter is ligated and sequenced. Since this is a long read sequencing platform, the Minnow program, which is the control software, discards the short reads. Therefore, previous attempts to directly sequence tRNA was not successful due to the loss of reads. So, also, last 15 nucleotides at the 5' end are not sequenced due to high translocation speed. So standard direct RNA sequencing has low yield in tRNA sequencing, and because of that, a new method is required to directly sequence these tRNA molecules. In the current study, there are three different library prep methods were used. In the first method, 3' end of the tRNA was polyadenylated to make the structure similar to mRNA. Then RNA adapter was ligated to the 5' end to increase the length of 5' end. Next, ONT library prep procedure was followed by ligating reverse transcriptase adapter and RMX adapter. But this method also has poor sequencing yield and low uniquely mapped reads. In the second method, instead of polyadenylation, pre-annealed tRNA and RNA splint adapter was ligated. However, DNA to RNA ligation at the 3' end is not so efficient. And next, ONT RMX adapter was ligated to the 3' end, but there is no reverse transcription step in this method. This method also yielded low sequencing yield and low uniquely mapped reads. In the third method, or nano-tRNA-seq method, 5' RNA and 3' RNA DNA splint adapter was ligated. Using a DNA-RNA hybrid adapter at the 3' end that improves the ligation at both ends, one end to the tRNA molecule and the other end to the reverse transcription adapter. Then ONT reverse transcription adapter was ligated and the reverse transcription was carried out. Finally, ONT DRS adapters are ligated and sequenced. Using this improved nano-tRNA-seq method, more than 200,000 base call reads were obtained, and this is up to 4-fold higher relative to previous methods. And nano-tRNA-seq method improved both 5' and 3' end coverage in both synthetic and biological tRNAs. Synthetic tRNAs are in vitro transcribed Drosophila melanogaster and Streptococcus pneumoniae tRNA, and biological tRNA is from yeast. And this method is highly replicable with native tRNAs. Alignment of native tRNA reads is challenging because the reads are short and also contain modifications. These modified bases cause inaccurate base calling, which leads to inaccurate alignments. So here in vitro transcribed Drosophila melanogaster mitochondrial tRNA for alanine and native Saccharomyces cerevisiae phenylalanine tRNA are shown. The long read mapper Minimap2 with recommended settings failed to align any biological tRNA reads. So BWA aligner for short read alignment was used with recommended parameters and also with custom parameters. As shown in the previous slide, Minimap2 aligned only 2.56 of total reads. In contrast, BWA aligned more reads with recommended settings, and relaxing the alignment parameters increased the number of reads mapped, but at the same time false alignments are also increased. So BWA mem alignment with specific parameters showed optimal alignment, which is 54.63% of total reads are aligned with 0.19% false alignments. I think you may remember the adapters that were ligated to the both ends of tRNA during the live preparation. Absence of these adapters have modest effect on in vitro transcribed tRNA, but has a major effect on biological tRNAs. Extending the tRNA molecules with RNA adapter is essential for guiding the correct alignment. As you see in this figure, especially at the 3' end, it must have a 3' end extension in biological tRNAs, and at least 25 nucleotides should be there in the 3' end. Native tRNA sequencing yielded low number of sequenced reads due to poor clogging and discarding short tRNA reads being classified as adapter-only reads. To avoid the read discarding, the custom MINAO configurations were used. Under default settings, adapter read time was 5 seconds. This means any read below this time is discarded without base calling. So adapter read time was reduced from 5 seconds to 2 seconds, and strand time 2 seconds in default settings was reduced to 1 second. Under these configurations, 12-fold more base call reads and 4.5-fold more uniquely mapped reads were obtained. Under default configurations, long tRNA with variable arms are preferentially captured, but under custom configurations, short tRNAs are also captured in increased amounts, and a significant increase in short tRNA were observed. So the relative proportion of the tRNA reads are better represented. For both synthetic and biological tRNAs, observed tRNA abundances are correlated with expected values under these custom configuration settings. The next approach for reduce clogging is linearizing tRNA by reverse transcription. Reverse transcription reduces clogging, stabilize translocation speed, and increase total yield. However, number of base call reads are 1.5 times higher without reverse transcription. This is due to nonlinear tRNA move more slowly through the pore, and they are highly likely to be recognized as reads due to the longer time. However, relative abundances of ISO acceptors are not affected by with or without reverse transcription. Nano-tRNA-seq was compared with 3 Illumina-based methods available in the literature. Correlation varies depending on the methods, and highest correlation was with MIM-tRNA-seq, which is 0.626, and that is not so high. Due to the fact that fragmented tRNA with modifications are subjected to PCR bias, and mapping fragmented samples cause spurious tRNA counts, and also there is substantial differences in Saccharomyces cerevisiae growth conditions in 2 occasions, and also there are differences in library prep and analysis methods. So these kind of differences is the main reason for observing this low correlation. In the literature and as well as in this study, it shows that base calling errors and mismatch to reference can be used to detect base calling modifications. This is obvious as biological tRNAs have more mismatches than in vitro transcribed tRNAs. Also, these mismatch positions are coincided with non-tRNA modifications. To check whether these mismatches are observed due to nucleotide modifications, nucleotide modifying enzyme was knocked out and studied. PUS4 enzyme that converts uridine 55 into pseudouridine 55 was knocked out. The wild type strain has mismatch at position 55, and PUS4 deficient strain showed correct match at U55 position, and other positions are not affected. tRNA modifications are introduced in sequential order. Pseudouridine 55 positively influenced the introduction of methyl at uridine 54 and adenine 58. In PS4 deficient strain, decrease in base calling errors at 55, 54, 57, 59 positions were observed. Further, these were confirmed with LCMS analysis showing significantly decreased methyl 5-uridine and methyl 1-adenine modifications in PUS4 deficient strain. Finally, effect of heat and oxidative stress over tRNA modifications and tRNA relative abundance was studied. Heat stress has significant change in abundance of one isoacceptor, and oxidative stress has significant change in abundance of 7 isoacceptors. However, modest difference in tRNA modifications was observed upon stress exposures. Upon heat stress, a substantial increase in base calling error frequency at position 76 terminal A was observed. Reduced coverage was observed at this position relative to its neighboring bases, and the significantly high deletion of terminal A for each isoacceptor was observed under oxidative stress. In summary, nano-tRNA-seq can be used for accurate measurement of native tRNA molecule abundance and their modification status at single molecule level. Increased total sequencing yield was obtained with extended 5' and 3' ends of tRNA, and increased base call reads and accurately aligned reads were obtained with custom sequencing configurations, and authors suggest that this tool will be useful for identifying biomarkers for cancer screening. And that's the end of for today's presentation, and thank you so much.